concept. Let's see if this works. There we go. All right. So, if you want to have a sort of technical definition of this, this concept, then basically pharmacogenetics is the study of variations in DNA sequences as related to drug response. And what does this mean? So this is kind of an oversimplification. The thought is that traditional medicine is what you see on the left side of the screen. In reality, it's never been that simple. We've always recognized, as I said, even back into the early 1900s, and even before that, that individuals are different. But traditionally, we can kind of say that our approach to patients has been sort of one size fits all. And so if a particular patient has a certain disorder, they're offered basically the same set of therapies. And you hope that a lot of these patients will benefit from those therapies. But there will be some patients who will not derive any benefit. And even worse, there will be some patients who might actually have adverse events or adverse effects as a result of that drug, including death in some cases. So the idea of precision medicine is that by using individual genomic variant data, we can then identify those specific individuals who would benefit from any specific therapy. And so then you can tailor the therapy to each individual patient so that every patient ultimately gets a drug or a therapy that is beneficial for that patient. And you hopefully avoid adverse events this way. All right. Now, how does this work in terms of biology? So we all know that we can have genetic variation. Some of these variants are, for example, those that might be in a promoter of a gene or in enhancer sequences or other regulatory sequences of the gene. And what they do is they alter expression of the gene, either transcription or and subsequently the translation and the protein product. And most of these variants will actually cause decreased expression, but some could actually cause increased expression of the gene product. And then other variants actually are coding variants that then can change the protein product or cause it not to be produced in a normal fashion. Now, what does that mean at the protein level? So drugs are ultimately interact with many different proteins in different ways. You can have drug metabolizing enzymes. So if you introduce a drug to a patient, then enzymes within that patient will metabolize the drug. And in most cases, inactivate the drug or cause it to be excreted. However, in some cases, the drug is actually a prodrug. So it's an inactive drug that is ingested by the patient or, and then becomes activated by the metabolizing enzyme. So you can have it go both ways. You can have some proteins that are drug transporters. For example, if a medication is ingested through the gastrointestinal tract, then there could be a transporter at the gut that causes it to be taken up into the portal circulation. Or you can have transporters at the cell surface of the target tissue, and they allow for the drug to be imported into the cell where then it's active. You can, of course, have a protein that is the direct target of the drug that can be affected by genomic variation. And you can also have other proteins that are actually not usually associated with the drug in any way, but when there is some sort of genomic variation that causes the drug, the protein to be changed or maybe overexpressed, and it starts interacting with the drug in an inappropriate way. That can lead to changes in the effectiveness of that drug. And then on top of that, you can actually have regulatory proteins that regulate any of these other proteins. And if you have genomic variation that affects those regulatory proteins, then that indirectly can affect the other proteins that, again, will cause a change in the effectiveness of the drug. Now, in the United States, we have roughly 1,200 drugs that are approved for clinical use, and about a tenth of them actually are affected by actionable pharmacogenes or genetic variation. And if you actually look at the prescriptions that are placed for different patients, about 4 billion are placed electronically, usually in the United States, and about a fifth of them are for drugs that have actual pharmacogenes associated with them. So this is, again, another list that you need to really read in or know in detail, but this is a list of different genes where variations in these genes can affect the action of a series of different drugs. And so this is one way you can kind of formulate the list. And this is the other way you can formulate the list, where you have different drugs and you associate them with variants in different genes that ultimately impair or change the activity of the drug, or the effectiveness of the drug. All right, so what I'm going to talk about in the next 15 minutes or so are first, how genetic variation can affect the dose of a drug that you need to give a patient. Second, how genetic variation might inform the actual choice of drug that you use for a patient. And then third, how genetic variation can actually help us design new drugs. And I'll give you examples of all of these. If we have some time, I'll talk a little bit about sort of more systems approaches in genomic medicine, and then how successful we've actually been in translating some of these concepts into clinical practice. And I'll leave you with a slide at the end with some resources on the internet for if you want to know a little bit more about personalized genomic medicine. All right. So let's start with drug dosing. So here's an example that will actually probably kind of go away in a while because this is a drug that is now slowly going to become obsolete, I think, for most patients. Nevertheless, it's still widely used, especially in my own clinical field, which is cardiology. So there are patients who, for a variety of reasons, you want to decrease their blood coagulability. For example, they may have a hypercoagulable state, or they may have certain cardiac arrhythmias that cause clots to form in their hearts that can break off and go to the brain and cause a stroke. So you want to give them an anticoagulant. And until recently, really the only anticoagulant we had for outpatients was something called warfarin, which is basically rat poison. And it inhibits a number of different coagulation factors that are kind of a cascade in the blood that ultimately lead to the formation of a clot. And this is kind of how warfarin works. It inhibits this protein, which is vitamin K epoxide reductase. And what it does is this enzyme actually causes the oxidized form of vitamin K to be reduced. And then this reduced vitamin K then actually activates a number of coagulation factors. And in the process, it becomes epoxidized. And then the enzyme causes the vitamin K to be reduced again. So it's a cycle that goes back and forth here. It turns out then that this is the direct target for warfarin. Also, warfarin can get inactivated by an enzyme called cytochrome P450-2C9. Now, it turns out that there are variants in the genes that code for these two proteins that can change the activity of this drug. And about 40% of the variability we have in the dosing requirements is actually due to variation in one or both of these genes. So it turns out that it's actually a very hard drug to initiate in a patient. You start out with a sort of guesstimate as to what dose they'll need, but then you actually have to do blood tests fairly regularly, often weekly or even more often, to figure out just how thin their blood really is and to adjust their dose. And it's really a very time-consuming, difficult process. And the problem is that if you undershoot and the blood isn't thin enough, then the patient can actually have a stroke, for example. Or if you've gone overboard and the blood is too thin, then the patient could actually bleed to death. So it's actually a very difficult medication to kind of manage, especially in patients who might not be that close to healthcare providers. So there were a number of trials that were undertaken to use genetic variant information from those two genes to kind of guide the provider as to what dose of the drug warfarin should be started on that patient. And the results were kind of interesting. So there was a European trial which showed that using the genotype information did increase the time that the patients were in the appropriate therapeutic window where their blood was appropriately thinned. And it was faster. They got to their target thinning basically much more quickly than patients where no genotype information was made available to the healthcare provider. Now, there was a US study that actually found no difference whether genotype information was available or not. So there was a bit of a difference between these two trials. And turns out the reason for this was that in the case of the European trial, the patients were mostly Caucasians, whereas in the US trial, the patients, 30% of them were African descent. And it turns out that in African populations or African origin populations, the variants that kind of determine the activity of warfarin are different from the variants that are more important in the Caucasian population. And so basically the wrong variants were being used to determine the drug dose that should be used in African-American subjects. And this is not an uncommon theme in genetics, as you know, where if you use the wrong reference population, you can end up getting spurious results or worse. And so, genetic background and racial variation is very important to keep in mind. Here's another example of where the choice of drug might be different depending on the patient's genetic variation. So another kind of blood thinner is called clopidogrel. This actually works on platelets, which are kind of enucleated cells in the blood that form part of the clots. And so clopidogrel is actually a prodrug that is activated in the liver, and then it goes and interacts with the platelet to help prevent clots from forming. And we use this, again, in my clinical field. Patients that are undergoing stenting of their coronary arteries, you need to use this, otherwise the metal stents in the coronary arteries will clot up and the patient will have a pretty large heart attack. Now, it turns out that there's a number of enzymes that will activate the prodrug to the active form, and there are actually genetic variants in some of the genes that encode these enzymes in the liver. And probably the most important one is in cytochrome 450 2C19, which has a number of variants that make that enzyme either pretty highly active or less active. And so if you have the variants that make the enzyme active, then that's fine, because then the patient can get the clopidogrel and it'll be activated and it'll work in the patient. But there's some patients that actually have variants that make this enzyme less active, and therefore if the patient gets the clopidogrel will not be adequately activated. And so again, in a situation like a coronary artery stent, that patient could end up having a large heart attack. And so if you know the patient is a poor metabolizer and has these variants in that gene, then you actually use an alternative drug instead of clopidogrel. All right. How about where you can use genetic variants to determine whether you should use a drug or not because of adverse drug reactions? So this is actually a pretty important problem. You can have a couple of 100,000 patients dying every year in the United States because of adverse drug reactions. So it is important to recognize this problem and to avoid it whenever possible. I'll give an example in the world of, again, it's important in cardiovascular medicine. So cholesterol, of course, as you all know, is a major risk factor for cardiovascular disease. And we use drugs called statins, which is the most commonly used drug around, especially for controlling cholesterol levels. And the statins work in the liver cells to basically prevent cholesterol from being synthesized. And then the liver cell, because it doesn't have enough cholesterol, it actually upregulates a receptor called LDL receptor that pulls in cholesterol from the blood, and that's how the blood levels of cholesterol will go down. Now, it is associated, some of these statins are associated with something called rhabdomyolysis, which is extreme muscle damage called rhabdomyolysis, and can actually be potentially lethal. And so it turns out that there's a transporter in the liver cell, the hepatocyte, called OATP1, B1, organic anion transporter protein, that allows for the statin to be imported into the hepatocyte. And there's actually variants in the gene that encodes this transporter that can make this transporter less effective. And if that happens, then more of the statin stays in the blood and doesn't get imported into the liver cell. And if it's in the blood, it then can basically attack the muscle cells and cause rhabdomyolysis. So it is actually important to see if you can recognize whether a patient has this variant. So it's a variant that actually is a coding variant that changes the activity of that transporter. So if you do, one of the statins that's most commonly associated with this adverse drug effect is called Simvastatin. And so if you actually are able to get genotype information, you can decide if the patient has a poor transporter, which would be the C variant, either in the heterozygous or homozygous variant, then you may consider either using a lower dose of the statin or actually using another drug in that family rather than the Simvastatin here. So here's where drug genotype information can give you guidance as to the dose or the choice of drug you want to use. Genotype information is actually very commonly used in cancer medicine as well, in oncology. And I'll give you one example here. So BRAF is a protein that is associated with a pathway that leads to cellular proliferation. And there's a somatic cell mutation in BRAF that causes those cells then to become proliferative. And this is actually a major cause of melanoma. And so there's actually a drug that will inhibit the activation or the activity of this mutated protein, but only when this protein has this specific mutation. So it won't work in every kind of melanoma, it won't work for every kind of mutation, but it works for this specific mutation. And when in a clinical trial, patients that had this mutation and received this mutation-specific drug had a longer-term survival. So this is time, and this is the patient surviving. And so more patients survive longer if they got that drug compared to the standard therapy at the time. And let me give you one more example here where at the population level, genotype information may be helpful. So we're going back to the drugs that are related to cholesterol inhibition. So there's a good form and a bad form of cholesterol. And so HDL cholesterol is actually a good form of cholesterol that protects you against cardiovascular disease. And there's a drug called delketrobib that increases the good form of cholesterol in patients. And so the hypothesis was, in this particular trial that was published in 2012, that increasing the good cholesterol should reduce the number of cardiovascular events in this population. And it turns out then that they didn't find a difference. So patients that got either placebo or the drug, there was no difference in the event rate for heart attacks and strokes and cardiovascular death. So the investigators didn't give up quite there. They did a GWAS study in their patients in the trial and identified a variant here that seemed to be associated at the genome-wide level, significantly associated with adverse cardiovascular events that were getting this active drug, delketrobib. So what we found out, or what they found out, was that patients that were homozygous for the minor allele, this is a this is a variant in the adenyl cyclase gene, non-coding. The patients that were homozygous for the minor allele actually, and they got the drug, they actually had a lower event rate than patients that were heterozygous or homozygous for the, the wild type allele. So it looks like that in this population, in a small subset of patients that actually are homozygous for this variant in adenyl cyclase, they actually do respond to the drug. And so what they're doing now is another trial that just has the design paper was just published earlier this year. And they're actually taking patients that have had a recent cardiovascular event. They genotype them to see who's homozygous for the minor allele, and then they go ahead and then randomize them to the drug with placebo. All right, we're going to be running out of time soon, let me just tell you a little bit about how genomic information can be used to design drugs, and then we'll just wrap up and leave some time for questions. So cystic fibrosis, as you may know, is the most common autosomal recessive lethal disorder in Caucasians. And it's due to variants in a gene that encodes for a chloride channel that's present in the lung tissue as well as other tissues. And it used to be pretty lethal. There's many different variants in the gene. Some of them cause the protein not to be produced properly. Some of them cause the protein not to get processed or upregulated to the cell surface. Now it turns out then that a number of different investigational drugs have been introduced, and one has now been approved and is in use, called ivacaftor. This is a drug that keeps this chloride channel open. So these mutations basically cause the chloride channel not to work, not to stay open. So this drug works in keeping the channel in the open state, but it only works for certain variants. So most commonly the G551D variant, which is actually a very small proportion of patients that actually have that variant, but if they have it, then this drug works in them. So here's a situation where a drug has been designed specifically for a variant, and only patients that had that variant actually will benefit from the drug. All right, let's go on. Let's skip over some of this because we're going to be running out of time. It's been a little bit tough to introduce a lot of these topics into clinical medicine. There's a variety of different players that are involved in making this actually work. And so obviously you need the research to be done and funded. But when there's evidence that certain genomic variants can actually inform clinical care, then you need to have test developers who can develop the clinical genomic test for it. You need drug manufacturers to design the drugs that will be informed by the genomic information. And of course, you need investors to invest in making this happen. You need regulators to approve this. You also will actually have insurance carriers that have to approve the use of genotyping and or the drug, that, especially if it's a novel drug designed for a specific genetic variant. And ultimately, of course, you need healthcare providers to buy in and patients to buy into this. And to give you an example, it doesn't work so easily. So this is a study out of Hong Kong looking at a number of different drugs that are used for epilepsy. And there is carbamazepine, which is a pretty widely used drug for epilepsy, has a potentially lethal side effect, causing a major problem with basically skin necrosis. And it is due to patients that have variants in this gene, HLA-B15. And they're at high risk of having this skin disorder as a result of getting carbamazepine. So Hong Kong, the government actually mandated that all patients starting in September 2008 be genotyped for this variant. So what did neurologists do? They decided that they couldn't be bothered doing genotyping. So in orange here are the number of prescriptions for carbamazepine. And as soon as September 2008 came, there was a drop in the number of prescriptions for this drug. And instead, physicians started using other drugs that may or may not be as effective. So they just couldn't be bothered doing the genotyping because they didn't really understand it that well. So these are some of the challenges that arise from trying to translate what we know theoretically to clinical medicine. All right, I'm just going to have this last slide in here. It gives you a list of different resources that can be used to learn a little bit more about pharmacogenetics and genomics and personalized genomic medicine, and how it eventually could be translated to clinical medicine. So I'm going to stop here and give us a few minutes in case you have questions. I think we're going to have people put in some questions by chat. So let's see if there's anything that's come up. So we have a question here. What would make this field more attractive to investors? That's a good question. I'm not a finance person. You know, I'm a physician and scientist, so it's kind of hard for me. Sometimes, you know, we're not actually as well trained as we should be in sort of getting, you know, industry funding sometimes. I think it's a matter of, one, it's a matter of educating potential investors as to what the potential utility would be, you know, at the population level. You know, we're not going to be talking about individual patients here. We're going to have to show that it will be relatively easy to do and cost effective, and can potentially help a large population, either, you know, in terms of genotyping or using the drugs. So if you can make that case that it's relatively easy, it's going to be cost effective, it's going to be applicable to a large population, then it, you know, could be potentially a profitable endeavor for them. Second question is, how long do you think it will take for this to become part of clinical medicine? So some of it, as you know, I gave you some examples where it has moved into clinical medicine. And one of the examples is ivacaftor, which is a drug that is now clinically available and being used for cystic fibrosis. The problem, of course, is cystic fibrosis itself is a relatively uncommon disorder. I guess it's the most common recessive disorder in Caucasians, but still, at a worldwide level, it's relatively less common. And then on top of it, ivacaftor works only for patients that have you know, variants that are actually pretty rare in that patient population. So while it is out there in clinical medicine, it's still going to be a relatively niche product. And then there's other examples. So I think the biggest and most commonly used area right now is actually oncology and cancer medicine, where not germline variants, but actually somatic cell variants. You know, if you genotype the tumor, identify the somatic cell variant that underlies the particular cancer, that's where it's being used to guide novel therapies that are actually being used in clinical medicine. So some of this has actually moved into clinical medicine. It's still somewhat limited. In other areas, it will be a matter of you know, cost effectiveness, showing all those different players that it actually makes sense to do genotyping. In some cases, you know, it may just be easier to use other drugs that don't have any known sort of actual pharmacogene. So I can't give you an answer as to, you know, when this is going to become widespread in many different facets of clinical medicine. It's present right now. It's being used, but, you know, wide scale, you know, maybe a decade perhaps, but that's just a guess. Take one or two more questions. I'm going to be running into Professor Prince's time here. What is the prospect of pharmacogenomics in medicine? Okay, well, we kind of addressed a little bit. What do we need to promote its clinical translational in medicine? Again, I think this is going to be a group effort. You know, we have many different players involved. So we need to have, you know, obviously the scientists, the healthcare providers involved, but also industry, regulators. And one of the ways to do it is to work with professional societies, work with your regulatory agencies in your country. And sometimes there are times when the FDA, for example, will convene a conference of interested parties. You sit down in a room, you know, pre-COVID era and hopefully post-COVID era. You have to sit down in a room and actually have discussions and panels and try to bring this to fruition. And then when you have buy-in from a large number of players, then, you know, you can make things happen more quickly. All right, I'm going to stop here. If you have other questions, you know, please let Heather know, and I'm happy to address them offline later on. Okay, we'll get, I think I've stopped sharing my screen. I'm going to stop my video and let Professor Prince start.
Great, thank you so much. So I will share my screen and get started. So let's see, if you can't see that, let me know, but I think we should be all set. So I'm going to switch gears a bit and talk about the ethics of personalized genomic medicine. So to give you a little bit of background, I'm a lawyer and I work in what's called the ELSI field, which is the ethical, legal, social implications of genetic testing, so ELSI. And I look at how different social actors, like an insurer, might use genetic information, but also how we think about who has access to data and what some of the other concerns may be. So today, what I want to do is really think very broadly about what some of the implications are for bringing this personalized medicine into clinical care. And so I'll cover three big buckets. I could spend hours talking about the intricacies of each of these. So I'm really just trying to give a little bit of a whirlwind view of some of the discussions that happen in the LC world and amongst clinicians as well about the implications of all of this. So the first big category to think about is data, right? In order to learn about all of these various drug interactions, as Ferhaan was talking about, or other genetic indications, we need data on hundreds of thousands of people, right? To learn the associations between drug and genetics, or genetics and disease. And so one of the questions then is, who owns that data, and what are the sharing implications, right? So oftentimes the data will come from clinical care or come from a research project. And so if I'm an individual who has consented to be part of a research study on pancreatic cancer, and then that goes into a biobank, and I get consented to do that, right? I have my permission, I understand what the risks and benefits are. But if that data is then shared for these bigger projects, then it's harder to know what happens to the data and who should own it. In the US, there are some states that actually have laws that say genetic information is the property of the individual. There's some global communities who say that genetic information is a cultural thing, right? And so there's the group genetics is owned by the country. And then there's just the practicalities of whether any of this information can be shared or sold. And so a lot of this depends on what the agreements were, right, in the consent documents, but it does have a lot of implications for what can be done and what the fairness issues are when people, if people understand how their data is used. Another category of questions that occurs around data and personalized medicine is, how does that get reincorporated back into medical records? So if we're really doing a lot of personalized medicine, right, we'll need to do more and more sequencing. Maybe it's something where everybody should be sequenced and just have their genetic sequence in their medical records. And so that physicians can go and find that information. Maybe it's something where every time, right, the more pharmacogenetics, for example, that we get, the more times somebody might have to do a genetic test in order to understand the implications. And so there's a lot of debate right now about what we put in the medical records, how accessible it is, and ways to make it more user-friendly. And so there's different mechanisms, like having a pop-up screen come up so that when a certain drug is prescribed, it says, hey, did you see that the patient has this genetic variant, or did you check whether this patient has this genetic variant, to encourage physicians to do that type of thing. But that's complicated, right? It's complicated to store the large amounts of data. It's a question of how much information gets placed into the medical records and how accessible it is, right? If it's just a PDF of a genetic sequence, then that's not as helpful for clinicians. And then there's this question of what's called interoperability, which is basically saying, can the medical records of the hospital that I'm at speak to the medical records of somewhere else, right? And so if I, as a patient, get genetic testing done here in Iowa, and then I am in California visiting family and need something, right? Then can the medical records talk to each other and have the information flow? Or if you need your family members' information, right, because of genetics. And so those are two big questions about medical records and how that helps to encourage personalized medicine, right? And then I hinted at this with the who owns the data, but there's a big question about secondary research. So in the US, there are requirements for consent. So if you're going to do genetic research with somebody's information, you have to get consent. However, there's an exception if it's a de-identified sample. So if the name is taken off and there's not a way to link the person to their genetic sample, then we can pool all of those samples together to create these biobanks and to have research done on the biobanks. But that can have some ethical issues, right? There are some research studies that people find don't match up with their moral beliefs, right? Or were comfortable with the information being shared in one way, but not another. And so it's hard to trace that research, and so there's questions in the LC world about how we do that the right way, right? Obviously, to make personalized medicine be something that we can have, we need hundreds of thousands of samples, but how do you balance that with proper consent and respect for people whose data we're using? Another question with that is whether or not we can actually de-identify something truly, right? And so in clinical or in personalized medicine research, it's really important to have not just genetic information, not just the genomic data, but also the phenotypic data. And the more data you have in, the better the prediction is, but also the easier it is to try to figure out, oh, this is somebody who was a patient of this age at this hospital who had this phenotypic information. And so it theoretically is possible to identify somebody. And so what does that mean for our ethical norms if our ethical norms are grounded on this idea of de-identification? So that's something that is talked about not just in genetic research, but also in just big data research in general, or implications of all of the data that is out there in the world. And so something that is talked about quite a bit in the LC world. Another question, and I saw in the chat, somebody brought up this as well, and then Ferhaan talked about it a bit, right? But there was also questions about what data we're using, right? And right now, there's a lack of diversity in some of the main biobanks that are used. The UK Biobank, or 100,000 Genome Project, that I'm gonna get the name wrong, but some of those big biobanks, right, are predominantly people of European ancestry. And so the clinical implications of that then are that those without European ancestry are more likely to receive uncertain results, or it's unclear, what the implications would be. So either they'll get a variant of uncertain significance, or there might be this variant, but we're not quite sure what that means for the medical implications. And so this is a really big justice question, right? For ethicists, we say, well, how are we then perpetuating some of the disparities, the health disparities that are especially relevant, unfortunately, in the US, right? What do we do to fix that bias in the data so that we can not perpetuate existing healthcare disparities. The hard thing is it's not as simple as just saying, well, let's get more people into the system, because one of the reasons that there's not a broader diversity of samples in the system is because of, you know, historic lack of trust of research and medical care, because of historical atrocities really that happened to various groups, right? And so there is understandable lack of trust. There needs to be relationships that are built among community actors to get more people into biobanks in a way that's respectful, whether it's, you know, a certain tribe and tribal norms. And so it's not just this on-off switch where we can pretty easily say, oh, well, let's get more people into these biobanks. We have to do it in a thoughtful, respectful way, but we really need to do it so that the justice implications on the back end mean that we have equal and equitable access to the benefits of precision medicine as well. So right now, the early adopters in the system are seeing are seeing greater accuracy, right, in how we can assess their information. Okay, so that is some of the data implications. So let me turn and talk a little bit about some of the cost implications. Like I said, this is just my whirlwind overview of some of the things that are talked about in this area. So this was mentioned a little bit as well today, but one of the questions is, who is going to pay for this? So if we're at the cutting edge of trying to determine when a genetic test result is going to change, you know, is going to have clinical utility in a way that shapes cancer care or in a way that shapes prescriptions, right? Unless clinical utility can be proven, insurers aren't necessarily going to cover it. And in the US system, we need insurance coverage in order to have access to care for the most part. But even in countries that have more of a public version of paying for healthcare, right, a more universal healthcare system, this is still a question. And so it's really important to prove the clinical utility, but sometimes you have to implement things in order to build the evidence base with enough people. And so getting insurance coverage at the front end of this is sometimes difficult. So one example is right now in the country, BRCA testing for breast and ovarian cancer is pretty widely covered for women and men who have a family history or some sort of symptoms or family history of symptoms. But if you get a negative BRCA test, then what might happen is you say, okay, well, let me get a panel test, right? Let me test across multi-genes that have implications for cancer. And sometimes those multi-gene panel testing aren't necessarily shown to be clinically useful, or they don't have the proven clinical utility for wide populations necessarily. They're starting to because they've been used more. But early on in the system, insurers said, we don't know that we're going to cover the cost of the panel test because we don't see the evidence for it yet. And so it can be a barrier to widely implementing something into clinical care. And then I think another question for all of us to ask ourselves is, you know, genomics is very expensive to do. The research is expensive to do, and I'm all for it. And I think it's fascinating, and we've come up with some really important measures. But I do think that it is something that everybody has to ask of, is this where we want the healthcare priority to be, right? We know all sorts of things that we can do from a public health perspective to improve the health of our society, right? Improve drinking water quality, stop smoking, access to care, and those sort of things are sometimes funding is cut for it. And so from a global perspective or a sort of top-down perspective, it is important to take a step back and say, okay, precision medicine sounds like wonderful, right? But how do we balance the cost that goes into developing those, and are we losing out on other things and other important measures that we could be taking from a public health perspective. And can we do both, right? I think is the real question of can we fund these systems in a way that helps us access both at once, right? So I don't think it's necessarily an either or, but it is something to consider. And then along with that is an access issue, right? So especially in the US, we do not have equitable access to healthcare. There are certain populations that are uninsured. There are people with all different types of insurance. Some insurances are going to cover the cutting edge technologies, some are not. And so if we don't have equitable access to precision medicine, then what does that mean for justice and for society? And it's not just about insurance coverage, right? It also can be about just being able to get to a doctor's office, right? So if you live in a rural community and you don't have a hospital that does cutting edge research right next to you, then maybe you aren't able to access these, to access this in the same way, right? And so thinking about the implications of that is really important and goes to cost because it takes money to fix access a lot of times. And then the third category that I want to talk about is this broad category that I label as information. And so one piece of that is privacy. So I talked about re-identifiability already, and that is a concern. But privacy is a broader lens than that, right? And so a lot of people are worried about not necessarily what happens to their information within clinical care or within research. There are many, many people who, even if you said we could re-identify your sample, would say, no worries, I still want my sample to be part of that biobank. I think that's important, right? But there's also evidence that people across the globe, really, that where this has been studied, have said, well, I don't want to participate in genetic research or to undergo medically recommended genetic testing because I'm worried about the implications outside in other areas of society. So in the US, one of the big debates right now is whether or not life, long-term care, and disability insurers would be able to use the information, right, to use a genetic test result to deny somebody insurance or to increase premiums. For the most part in the US, those types of insurers can use that information. Florida just actually passed a bill and it went into effect in July that stops those 3 insurers from using genetic information. And so there is some movement in Florida. There's also been some recent really big movements in this anti-discrimination area in Canada and in Australia. And so this has been a debate across the globe. And if you're interested in learning more about sort of globally some of these protections, there's a group called the Genetic Discrimination Observatory. I'm one of the members of it, but it's an international collaborative group that looks at genetic discrimination laws and policy across the globe. And we're just getting up and running, but there's some really exciting collaborations from that project. So I jumped ahead to this genetic discrimination part. I should also say that in the US, our main framework is called GINA. This is the Genetic Information Non-Discrimination Act. And so GINA was passed in 2008, and it is a federal law that prohibits health insurers and employers, subset of those. It doesn't cover all employers, but covered employers and health insurers are not allowed to take into account genetic information in hiring and firing and pay decisions, or from the health insurance perspective, in accepting or denying insurance or premium amounts. And so GINA has been going on for over a decade now and is protective in those areas, but it does have the gaps, right? And that's where people are still concerned about, and where I mentioned the life, long-term care, and disability insurers. And then another big question, and this goes really well to Ferhaan's study that he showed where there was a policy that said, well, now you have to undertake genetic testing, and the provider said, oh, never mind, I'm not going to do this, right? And so there is a question about how much healthcare providers have either the ability to understand this information and how much of a burden it is, right? And so not all healthcare providers are trained in complex genomics. Maybe they know a bit about genetic testing, but they're not comfortable discussing those implications, right? Or it's a, one type of doctor and one type of specialty, but the information they get has implications for another specialty. And oftentimes, right, in medical systems, you can get a consultation with another doctor, and that's fine. But there are a lot of healthcare providers that work in solo practices or in small practices or in rural communities that might not have that ability to understand or to get the resources that they need in the same ways. And even if they can, the time commitment and the burden on learning that information and figuring out a way to easily communicate the risk information to their patients is a burden when we already have a system where you might have 15 minutes in front of a doctor and that's it, right? For the issue at hand. So there's a lot of other time burdens and cost burdens and all of that competing. And so what is the burden on healthcare providers of precision medicine? And so I'll leave it at that, but I'm happy to take some questions, and I will stop sharing my screen so that I can read any questions that might be coming on. Great, so I see a few have come in. So one is, as far as education and personalized medicine is concerned, are we doing enough in this area? And I think we're doing more, right? One of the questions is, who are we educating, right? I think there is, more education about genomics and genetics and personalized medicine in medical school and residencies and that type of thing. And so that's good. And in conferences, right, there's continuing education that covers this area. There's also a huge growth of genetic counselors, right? So these are, in the US, they're master's levels, right? So they have a 2-year program after finishing undergraduate school. And their job, they sit sort of in the middle of patients and communication and the counseling side and the genetics side. And so that group is remarkable at educating the public and talking about the risks and benefits. But there's not that many of them compared to how much genetic testing we would need to do to really get personalized medicine there. So I think there are some efforts and it's growing. But on the other hand, the lay public, doesn't necessarily know a lot about this, and it's a lot to learn, right? And so thinking of ways to help patients make those decisions, to help them understand the implications, to understand risk, is something that we need to continuously work on. So another question is, do I feel that privacy and data laws hinder the development of this field of medicine? And so I think we can think about this a couple of different ways, right? On the one hand, privacy and data laws do end up restricting researchers, right? And in some ways, right? But without those, you might not have any subjects, right? Because people don't want their research used or their data used in a way that doesn't protect them. So you have to find the balance between allowing research to go forward, but doing that in a way that respects human rights, that gives consent and all of that. And one of the things I use as an example quite a bit is there's a lot of studies that have been done where potential participants are asked, would you be comfortable with X, Y, or Z research done? And what a lot of those studies have found is that the number one thing that people want is to be told what's going to happen with their data, and to be asked if that's okay. And then many will say, yeah, I'll join that biobank. I will do this, right? And so do the privacy laws hinder the development? It might slow things down a little bit because you have to ask permission from people, but I don't think it slows it down so much because people are generally willing to participate as long as they're respected and understand what goes on. And the privacy laws can, in fact, help more people to feel comfortable to participate in research in that way. So I hope that answered your question, and I think we're just about at the hour. So as Ferhaan said, I'm happy to answer other questions. If you would like to email Heather, I'll make sure to get back to you. And thank you all so much for coming.
Thank you both. I appreciate it. And everyone, I'll be emailing out the video this afternoon along with the survey. And next week, we will be emailing all of you with the opportunity to participate in a massive online open course for the topics in human genetics. So look forward to that. And everyone take care. We'll see you soon. Thank you.
Bye. Thanks, Heather.